It is reasonable to suppose that natural selection operates to keep genome size as low as possible. After all, every time a cell divides, it must replicate all its DNA. The more it has to copy, the greater the room for error, and the more energy and time the process requires. It's quite an undertaking for the amoeba, or newt, or lungfish. So what could have caused the amount of DNA in these species to get so out of hand? In cases of unusually large genomes, we can only infer that some other selective forces must have negated the selection-driven impulse to keep the genome slim. It could be, for instance, that large genomes are advantageous to species likely to be exposed to environmental extremes. Lungfish live at the interface of land and water, and they can survive protracted periods of drought by burying themselves in mud. It could be they need more genetic hardware than a species adapted to a single medium. Two major evolutionary mechanisms account for this DNA excess: genome doubling and the proliferation of particular sequences within a genome. Many species, particularly in the plant kingdom, are actually the product of a cross between two pre-existing ones. The new species often simply combines the DNA complement from each of its parent species, yielding a double genome. Alternatively, through some kind of genetic accident, a genome may get doubled without input from another species. For example, one of the standbys of molecular biology, Baker's yeast, has about 6,000 genes. But close inspection reveals that a large proportion of those genes are duplicates. Baker's yeast often has two divergent copies of many of its genes. At some early stage in its evolutionary history, the yeast genome apparently got doubled. Initially, the gene copies would have been identical, but over time they have diverged. An even richer source of excess DNA has arisen from the multiplication of genetic sequences capable of replicating and inserting themselves at more than one site in a given genome. These so-called mobile elements have been found to come in many varieties. But when their discovery was first announced by Barbara McClintock in 1950, the very idea of jumping genes was too far-fetched for most scientists accustomed to the simple logic of Mendel. McClintock, a superb corn geneticist, had already endured something of a bumpy career ride. When it became clear in 1941 that she would not be granted tenure at the University of Missouri, she came to Cold Spring Harbor Laboratory, where she would remain an active member of the staff until her death in 1992 at the age of 90. McClintock once told a colleague, "Really trust what you see." This was exactly how she did her science. Her revolutionary idea that some genetic elements could move around genomes followed simply from observable facts. She had been studying the genetics underlying the development of different colored kernels in corn, and noticed that sometimes, partway through the development of an individual kernel, the color would switch. A single kernel might then turn out variegated, with both patches of the expected yellow cells and patches of purple ones. How to account for this sudden switch? McClintock inferred that a genetic element, a mobile element, had hopped into or out of the pigment gene. Only with the advent of recombinant DNA technologies have we come to appreciate just how common mobile elements are. We now recognize them as major components of many, if not most, genomes, including our own. And some of the most common mobile elements, those that appear again and again in different sites in the same genome, have earned names reflecting their itinerant lifestyles. Two fruit fly mobile elements, for example, are called gypsy and hobo. And among those who study a simple plant called volvox, one mobile element is honored for its extraordinary capacity to jump around the genome. It is known as the Michael Jordan element. Mobile elements contain DNA sequences that code for enzymes that through their capacity to cut and paste chromosomal DNA, work to ensure that copies of their particular element are inserted into new chromosomal sites. If a jump carries a mobile element into a junk sequence, the functioning of the organism is unaffected, and the only result is more junk DNA. But when the jump lands the mobile element in a vital gene, thereby disabling its function, then selection intervenes. The organism may die, or otherwise be prevented from passing on the new jumped-in gene. Very rarely the movements of mobile elements may either create new genes or alter old ones in a way that benefits the host organism. Over the course of evolution, therefore, the effect of mobile elements seems mainly to have been the generation of novelty. And, curiously, in recent human history, there is little evidence of active jumping. Most of our junk DNA, it appears, was generated long ago. In contrast, the mouse genome contains many actively reinserting mobile elements, making for a much more dynamic genome. But this seems not to trouble the mouse species unduly. The intrinsically high reproductive potential of mice likely helps the species as a whole tolerate the genetic disasters attending frequent jumps into vitally functioning genetic regions. Having been used to establish many of the basic facts about how DNA functions, E. coli's track record as a model organism was unparalleled. Not surprisingly, its genome therefore ranked high on the Human Genome Project's early to-do list. It was Fred Blattner of the University of Wisconsin who was most eager to start sequencing E. coli. But his grant proposals went nowhere until the HGP got funded, and he was awarded one of the first substantial sequencing grants. Were it not for his initial reluctance to adopt automated sequencing, his lab would have been the first to sequence a complete bacterial genome. But in 1991, his strategy for scaling up the operation was an old-fashioned one: employ more undergraduates. Another latecomer to automation was Wally Gilbert, whom I had urged 2 years before to have a go at the smallest known bacterial genomes, those of the parasitic Mycoplasma, tiny bacteria that live within cells. Sadly, when a clever new manual sequencing strategy of his came to naught, his Mycoplasma project died with it. Blattner did, however, accept automation in time to establish in 1997 that the E. coli genome contains some 4,100 genes. But the broader race, to complete the first bacterial genome, had been won 2 years before at the Institute for Genomic Research, TIGER, by a large team led by Hamilton Smith, Craig Venter, and his wife Claire Fraser. And the bacterium they sequenced was Haemophilus influenzae, from which 20 years earlier Smith, a towering 6'6" one-time math major who had gone on to medical school, had isolated the first useful DNA cutting, restriction, enzymes, a feat that won him the Nobel Prize in Physiology or Medicine in 1978. With Haemophilus DNA prepared by Smith, Venter and Fraser used a whole genome shotgun approach to sequence its 1.8 million base